Hi guys, because you missed lab today, I'm going to give you an overview of what I talked about both in the pre-lab lecture and then after everybody had drawn their reactions on the worksheet. I talked about the different structural features of a protein, so I'm going to go over that as well too. So lab this week was about amino acids. And I know that Dr. Smith went over this in lecture on Monday and Wednesday, so a lot of this should be familiar to you, but I just want to make sure that you're clear on all of the concepts so that you can do a good job on your worksheet and your quiz next week. So first off, every amino acid has the exact same what we call a backbone structure. So that is going to be a nitrogen that has two hydrogens on it, followed by a carbon. That carbon's got a hydrogen and another carbon bonded to it, and this second carbon has a double bonded oxygen and an OH group. And so the second carbon in the one that's bonded right to the nitrogen, that has to have something else attached to it as well. If you look, it's only got 3 bonds on it. So that other thing is actually what makes each amino acid unique. And so I know you guys don't like this, but I'm going to put R there, and all R means, remember, is anything. So it could be a hydrogen, it could be a methyl group, it could be any organic double bond O, OH, and glycine and alanine are different because instead of a hydrogen coming down from that alpha carbon, we have a CH3 group for alanine. So this is alanine, and its abbreviation is AL. Oops. ALA. Okay. Now, for the lab, what I've asked you to do is, and I'll showing you which two I want you to work with. You're actually going to react these together on paper to form a dipeptide, which is just two amino acids coming together. And so okay, so now if I'm going to react both glycine and alanine together to make a dipeptide, the reaction is really pretty simple. What ends up happening is you take the hydroxide, the OH group, off of the amino acid on the left. That goes away. One of the hydrogens on the nitrogen of the other amino acid goes away, and you end up forming a new bond between the carbon where you took the OH off and the nitrogen where you took the hydrogen off. And then, so if I draw that out, I end up with H2N C H C double bond O, and that one on the left is my glycine. And now here is my new bond between the carbon on the glycine and the nitrogen on the alanine. And so now I've got my nitrogen there, hydrogen, carbon, hydrogen, C double bond O, OH, and my side chain on that amino acid is a methyl group because that's alanine. And what I've just created is a dipeptide. So there are a couple of things that you should note about the structure of peptide chains or proteins. If you look, one side of my amino acid, the side on the left, ends with an NH2. And the other side ends in a C double bond O OH, a carboxylic acid group. We call the side of the chain that ends in NH2 the N-terminus. Kind of makes sense because it ends in nitrogen. And the part that ends in the COOH, we call the C-terminus. Would make more sense if it was the O-terminus, but that is not what people call it. It is the C-terminus. And so when we're listing out the order of amino acids, we always list it out, which we call that the amino acid sequence. We list them from the N-terminus to the C-terminus. Okay, the one other thing that we do need to note on this dipeptide is the bond that links the two amino acids together, which is right here. This is what we call the peptide bond. And the other thing that I do want to just kind of note, and I'm just going to highlight it, is we have formed a new functional group by linking those two together. The C double bond O N, that's one carbonyl group that we did not talk about in lab a couple of weeks ago. So when you have a C double bond O N, and then I promise you I won't use the R this time. That carbon obviously has something else bonded to it to make its fourth bond. And the nitrogen is also going to have two things bonded to it. But the C double bond O N is the amide functional group. AMIDE. And so every time you link two amino acids together, you do get an amide. Okay. So now it's time for you guys to go and do problem 1 on the worksheet, which means I need to assign you two amino acids to react together. So the first amino acid that I'm assigning you is going to be cysteine. And so cysteine is an amino acid that has a side chain, and I'll draw it down this way. This is that, in fact, I'll make it blue like I made it on that glycine. The side chain for cysteine is CH2SH. And so basically in place of the hydrogen, you're just going to draw that CH2SH group. The second amino acid that I am going to have you guys work with is lysine, which its R group is coming down. You've got a CH2, 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 CH2. Whoa, it doesn't really look like a C, does it? CH2, NH2. And so what I need you guys to do on the worksheet is go ahead and react. First, draw those two amino acids out, and then you're going to draw a reaction arrow and show me the dipeptide that they create. When you've got that dipeptide drawn out, you also need to identify the what does it say? It says place a circle around the peptide bond that was formed, label your N-terminus and your C-terminus, and oh yes, you also need to show the abbreviated form of the amino acid sequence. So just so that you guys know, the abbreviations for those two amino acids are, it is CYS for cysteine and LYS for lysine. Most of the amino acids, it's just the first three letters. There's a couple of exceptions though. So that is part one. Now for part two, what you're supposed to do is in lab, you guys created a dipeptide and you both drew it and you made a molecular model of it using our model kits. But then the group next to you also made a dipeptide. And just for some more practice with drawing this reaction out, because the more you write it, the kind of better you become at it, and the easier you find it, you guys were going to link your two dipeptides together to create a tetrapeptide. So I'm actually going to draw a dipeptide that you can pretend the people next to you had made that one out, so that you can practice making linking your dipeptide to another dipeptide for question number 2. So the dipeptides that I'm going to work with are valine and methionine. And so just to draw those out very quickly for you, the dipeptide that they would have had is NH2, CH. That one is a CH, and then that carbon has 2 CH3s bonded to it, and then C double bond O, N, H, C. Coming down off of that one is a CH2, CH2, S, CH3, and then we got our hydrogen, and then a carbon, C double bond O, OH. And so their 2 amino acids were, what did I call that one? That one is valine. The one on the right is methionine. Valine is VAL, and methionine is MET, and so its amino acid sequence, again, starting at the N terminus is valine, and then the C terminus is methionine. And so you are going to take the dipeptide that you created in part 1 and add this dipeptide that I've drawn to your dipeptide. You will write that out as a chemical reaction, so you'll draw the 2 dipeptides with a plus in between, and then a reaction arrow, and then the tetrapeptide that results, and also the amino acid sequence. So that is it for the worksheet. And now I do want to give you some information about the various structural features of a protein that lead to its 3-dimensional shape. And in fact, I'm actually not going to talk about it myself. Instead, if you look at the email that this link was attached to, you're going to see that I've given you instructions on how to get to a video that is on the Khan Academy website, because they actually did a very good job of describing the structural features. So go back to that email and pull up that video. You have to know the structural features. I promise you it will show up on the quiz for lab. So go ahead and do this. Hopefully you guys can turn in your worksheets to myself after class. If you do that, I will be able to get them graded to you and back in time to use for your quiz in lab this coming week. And if you have any questions, feel free to either email myself, Dr. Smith, or Dr. Griffiths, or stop by our office hours. My office hours are actually this afternoon, but I have no problem setting up a time to meet with you Monday morning or even sometime on Friday. Okay, so good luck, guys. Bye.